In Unit 3, Lecture 1, Part 2, we're going to be starting a new section where we're looking at virulence factors that promote bacterial colonization of the host. The remainder of Unit 3, Lecture 1 and all of Unit 3, Lecture 2 will deal with this topic. Again, virulence factors that promote bacterial colonization of the host, the ability of bacteria to establish themselves in or on the body. So in this SoftChalk lesson, we're going to be looking at the ability to use motility and some other means to contact host cells and possibly to disseminate within the host. As always, make sure you start out by reading your fundamental statements or bullet points for this SoftChalk lesson, and make sure you learn the detailed learning objectives that you'll be tested on for our Unit 3 exam. Also, beginning with this lecture, we're going to see some more highlighted bacteria like we did in Unit 1 that will appear on the exam. And the highlighted bacterium for this SoftChalk lesson will be Helicobacter pylori. So again, what we're looking at are virulence factors that promote bacterial colonization of the host. And we're eventually going to look at 6 different factors over the next this lecture and the next lecture. The ability to use motility, another means to contact host cells and possibly disseminate within the host. The ability to adhere to host cells and resist physical removal. The ability of some bacteria to invade host cells. The ability to compete for iron and other nutrients. The ability to resist innate immune defenses, and the ability to evade adaptive immune defenses. So again, in this lecture, we're going to look at the first of these factors, the ability of motility and other means to contact host cells and disseminate within the host. Now, mucosal surfaces of the respiratory tract, the intestinal tract, and the genitourinary tract constantly flush bacteria away to prevent colonization of host mucous membranes. They do this, of course, through the mucus they secrete. Motile bacteria, however, can use their motility and chemotaxis to swim through the mucus towards mucosal epithelial cells to eventually make contact with those cells. And many of the bacteria that colonize the mucous membranes of the bladder and the intestinal tract are motile. So we think motility helps these bacteria move through the mucus between mucin strands or in places where the mucus is less viscous or more watery. And there's quite a few bacteria that use motility, things like Helicobacter pylori, Salmonella species, our friend E. coli, Pseudomonas aeruginosa, Vibrio cholera, and such. And then once they do contact the cells, which we're looking at in this SoftChalk lesson, in the next lesson we'll look at how they can attach to cells to begin the colonization process. So this is an animation we showed you under bacterial flagella, showing how a motile bacterium can contact a host cell by swimming through the mucus. And the yellow here represents mucus, and the green dots indicate a gradient of chemoattractant being produced by the epithelial cells that form the mucous membranes. So the bacterium can use its motility and its chemotaxis to swim through the mucus and make contact with the epithelial cells of the mucous membranes. We also showed you this little movie of Pseudomonas. We'll show you a little bit of it, showing how it uses its flagella. We'll let that load in. So there we see the Pseudomonas with its single polar flagella. These are the pili on it. And the idea there is it's able to swim through the mucus and get down to the surface of the cells of the mucous membranes, where eventually it can first use pili and then cell wall adhesins in order to attach. And that goes on for a while. And there's a few movies we also looked at showing you the flagella of bacteria. Now, example we're going to look at here and in several of the other SoftChalk lessons coming up is the bacterium Helicobacter pylori. And Helicobacter pylori causes chronic gastritis and most duodenal and gastric ulcers. It also increases the risk for stomach cancer. And it has a whole lot of virulence factors going through it that we'll eventually be talking about. Can produce urease, an enzyme that breaks down the urea, a waste product, of course, of metabolism, into ammonia and bicarbonate. And the ammonia and bicarbonate act as buffers to neutralize the hydrochloric acid in the stomach. So by breaking down urea, it forms buffers to help neutralize the acid. The urease is also thought to alter the proteins in the mucus, changing it from a solid gel to a thinner fluid that the bacteria can more easily swim through by means of their flagella. So using motility and chemotaxis, the bacteria move away from the concentrated acid in the lumen of the stomach, a repellent, towards decreasing concentrations of the epithelium of the stomach. Urea is also thought to act as a chemoattractant, so it moves towards the urea being produced by stomach cells. And then eventually see how it can use adhesins to adhere to epithelial cells that line the epithelium of the stomach or of the duodenum. To further help the bacterium, Helicobacter pylori produces an acid inhibitory protein that blocks acid secretions by the parietal cells of the stomach. So it acts as a chemical that's going to prevent the stomach from releasing the epithelial cells of the stomach from releasing the acid. And then bacterial toxins can lead to the production of inflammatory cytokines and chemokines, as well as mucinase and phospholipase that damage the gastric mucosa. The cytokines and chemokines in turn trigger an inflammatory response, being inflammatory cytokines and chemokines, that causes neutrophils to leave the capillaries, accumulate at the area of infection, and through extracellular killing, discharge the contents of their lysosomes. Now, of course, that is done to kill the bacteria that have accumulated there, but of course, the same enzymes and such that the neutrophils produced to kill the bacteria are equally good at destroying the mucus-secreting mucous membranes of the stomach. It also secretes some toxins like cytotoxin A that damages host cells, causing them to release nutrients to feed the microcolonies of Helicobacter pylori. And once that protective mucus layer is no longer being produced because the cells, the epithelium and the cells producing the mucus are being damaged by the neutrophils, then that protective barrier is gone and the gastric acid can lead to ulceration of the stomach. And this is what leads to either gastritis or gastric and duodenal ulcers. And we have a little animation to kind of show you that process. Now, what we're looking at here primarily, again, is the motility part. We'll talk about the adhesins and the inflammatory response and all of that stuff as Unit 3 progresses. So in this animation, this is the mucus layer that's protecting the epithelial cells that line the stomach and duodenum from the acid or protons that from the hydrochloric acid. Now, Helicobacter pylori is actually a S-shaped or spiral-shaped bacterium with Bophotrichus arrangement of flagella, but I can't really draw that and show it moving very well, so I'm just going to show you a bacterium with a flagellum to indicate the general idea, although that doesn't look like Helicobacter pylori, but the same principle applies. So again, it's going to use its motility and its chemotaxis as it's attracted urea and other chemicals down here on the mucous membrane, and it's able to swim through the mucus to make contact with the cells, as we see here. So that's using motility to make contact with cells. And again, many bacteria that infect the intestines, the urinary tract, use their motility for that purpose, to be able to swim through that mucus layer and make contact with cells. Then cell wall adhesins allow the bacterium to bind to receptors on the host epithelial cells of the mucous membrane of the stomach or intestines to colonize and resist flushing. So we'll see in the next section that adhesins can allow the bacterium to bind to adhesin receptors. And that way it's able to adhere to cells and resist flushing. The bacterium then releases toxins that lead to excessive production of inflammatory cytokines and chemokines, as well as mucinase and phospholipase that are going to damage the gastric or intestinal mucosa. And of course, these cytokines and chemokines that are released are inflammatory cytokines, so they're going to trigger a local inflammatory response. So here we see some toxins affecting the cells, causing the release of inflammatory cytokines and chemokines. That's going to lead to vasodilation of local capillaries, making the capillaries more permeable. And that allows the neutrophils to leave the capillaries where they're chemotactically attracted to the area of infection up here. So again, as the neutrophils leave the blood vessels and crawl to the area of infection, at high cytokine and chemokine concentration, they're going to release the contents of their lysosomes for extracellular killing to kill the bacteria without ingesting them. But again, those same chemicals that kill the bacteria are equally good at killing epithelial cells of the mucous membranes. So there it is, discharging the lysosomes, and that kills bacteria, but it also winds up damaging epithelial cells. And so now when the epithelium is gone and the mucus is not secreted, the acid that was in the lumen of the stomach and the first part of the duodenum can damage the tissue underlying the mucous membranes, leading to ulceration and eventually bleeding, resulting in the bleeding ulcers. But again, what we're concerned with in this particular SoftChalk lesson is the first part here, where the motility helps the bacteria get through the mucus and make contact with cells. And there's our highlighted bacterium, Helicobacter pylori. When we talked about quorum sensing and individual behavior, we mentioned that individual Pseudomonas aeruginosa, what we call planktonic, rather than those that are in a biofilm, use their polar flagella to move through water and mucus to make contact with the body's mucous membranes. And then it can use pili and cell wall adhesins to adhere and eventually form a biofilm. So again, in figure 1, we see Pseudomonas using its polar flagellum to swim through the mucus, where eventually pili can first begin to adhere to pilus receptors for the adhesive tip of the pili. Now, another good example of bacteria using motility are the pathogenic spirochetes. Because of their thinness and their internal flagella or axial filaments, their corkscrewing motility, spirochetes are more able to penetrate host mucous membranes and skin abrasions in order to enter the body. But then the motility and the penetration may also enable the spirochetes to penetrate deeper in the tissue, where they can enter the lymphatics and the bloodstream and disseminate to other body sites to set up new infections. And that includes bacteria like Treponema pallidum, the bacterium that causes syphilis, Leptospira that causes leptospirosis, and Borrelia burgdorferi that causes Lyme disease. So remember, the spirochetes have the internal flagella or axial filaments, as we see in figure 2, that start at both ends of the bacterium and wrap around the spiral towards the center. And these axial filaments lie under the outer membrane but on top of the peptidoglycan and allow it to have its characteristic corkscrewing motility. So again, this is an animation we showed you back in Unit 1 showing how spirochetes can use their corkscrewing motility as well as adhesins and invasins to adhere to and drill through the endothelial cells that form capillaries. So they can enter the lymphatics and they can enter the blood through this manner. They can also use their motility to pass between the endothelial cells and enter the blood. So the spirochete we see on top is going to be using its motility to adhere to and then drill through the cell. And the one we see in a minute on the bottom is going to go between the endothelial cells and then be carried by the blood to another part of the body. So adhesins allow the tip to adhere where it uses its motility to drill through and enter the bloodstream. And as we saw, the one on the bottom down here is passing between the endothelial cells. And the blood then carries the spirochetes to a new part of the body, where they can use the same technique to leave the blood vessels and set up a new infection at a different site in the body by being able to pass through the cells or between the cells. So we know that the motility of spirochetes does help them to disseminate in the body as well as allow them to enter the body. And we showed you this movie again of the dark field microscopy of the spirochetes, where you can see the little corkscrewing motility there at the tip that allows it to drill through cells and between cells. Now, some bacteria that are not motile use some other techniques to spread within the body. Many bacteria produce enzymes like elastases and proteases that degrade the extracellular matrix proteins that surround cells and tissues, and that makes it easier for the organism to disseminate. So again, often this extracellular matrix that surrounds our cells helps to localize the infection. But if they can break down that extracellular matrix, then they're able to move to other cells. And a nice example of that is Streptococcus pyogenes that causes strep throat. This produces an enzyme called streptokinase, which lyses the fibrin clots produced by the body as part of the inflammatory response in order to localize the infection. So by activating the blood clotting pathway at an infection site, fibrin clots form that tend to trap the bacteria and localize it. But by breaking down the fibrin with streptokinase, they can then leave the clots and spread to surrounding areas. In addition, Streptococcus pyogenes produces DNase, an enzyme that degrades the cell-free DNA found in pus. And it's often the DNA in pus that's actually extruded by the neutrophils in the area that produce the viscosity of pus. And that again makes it harder for the bacteria to spread and localize it. But by breaking down the DNA, that makes the pus much less viscous, and the bacteria can then spread from the localized site to new tissue. Staphylococcus aureus, that often causes accidental and postoperative wound infections or abscesses, produces surface adhesins that bind to the extracellular matrix proteins and polysaccharides that surround our cells. That includes things like fibronectin, collagen, laminin, hyaluronic acid, and elastin. And Staph aureus produces proteases and hyaluronidases that dissolve these components of the extracellular matrix, and that provides food for the bacterium and again helps them to spread. So some bacteria may not use motility to spread, but they can produce enzymes and other such compounds that can break down the extracellular matrix that tends to localize the infection and stop it from spreading. And then along another line, the good and bad of diarrhea, we'll see later on under toxins that some bacteria produce toxins that induce diarrhea in the host. Now, diarrhea is actually part of our innate immunity. It's the way we flush out harmful microbes and toxins out of the intestines. So on one hand, diarrhea is advantageous to the body because it's flushing out harmful microbes and their toxins. But on the other hand, it's beneficial to the bacterium inducing the diarrhea because it also flushes out a good deal of normal flora of the intestines and reduces competition for nutrients between microbiota and pathogens. And diarrhea also enables a pathogen to more readily leave one host and enter a new host through fecal-oral route. Of course, with solid feces, the bacteria tend to be entrapped in that, but when you have a watery feces, as in diarrhea, it's much harder to clean up, it gets on everything, and it makes it much easier for bacteria to be spread via the fecal-oral route than it does with solid stool. So that's the role of motility in contacting cells and helping bacteria to disseminate. And as always, there's a little self-check quiz you can do to test yourself on this particular SoftChalk lesson.